Hello, welcome back to Regulatory Affairs 101. I am Nancy. Today, we are breaking down FDA's updated draft guidance on demonstrating substantial evidence of effectiveness for human drug and biological products. This is one of FDA's most foundational standards, and the new draft brings important clarity and flexibility for modern development programs. FDA's goal is simple: modernizes how sponsors can demonstrate substantial evidence of effectiveness. The guidance clarifies acceptable trial designs, data sources, and analytical approaches while reinforcing the statutory requirement for adequate and well-controlled evidence. It also highlights flexibility for innovative products, rare diseases, and complex biologics. The legal standard hasn't changed. Substantial evidence must come from adequate and well-controlled investigations. Traditionally, that meant 2 trials. But FDA now explicitly supports 1 pivotal trial plus confirmatory evidence as long as the totality of evidence is strong and reliable. FDA recognizes several control types: placebo, active dose response, and historical controls, though historical controls are still rare. Regardless of design, studies must have clear objectives, defined endpoints, appropriate populations, randomization, blinding, and a pre-specified analysis plan. A single pivotal trial may be enough when supported by strong confirmatory evidence that can include supportive clinical data, mechanistic or biomarker evidence, real-world evidence, or natural history data, especially in rare diseases. The key is that confirmatory evidence must be independent, reliable, and clinically meaningful. Examples include robust dose response, consistent subgroup effects, strong mechanistic rationale, well-characterized natural history, carefully justified external controls, RWE from registries, claims, or EHRS, and, when applicable, animal rule evidence. FDA encourages adaptive designs, Bayesian approaches, master protocols, and enrichment strategies. But these must include pre-specified rules, type 1 error control, and clear interpretability. Real-world evidence can support effectiveness, external controls, long-term outcomes, and generalizability. FDA expects fit-for-purpose data, transparent provenance, robust methods, and strong bias mitigation strategies. FDA accepts clinical outcomes, surrogate endpoints, composite endpoints, and patient-reported outcomes. Endpoints must be clinically meaningful, and surrogates need strong biological possibility and empirical support. For rare diseases, FDA allows greater reliance on natural history, external controls, intermediate endpoints, and Bayesian borrowing, but the evidentiary standard remains the same. Results must be reliable, interpretable, and not due to chance or bias. FDA evaluates treatment effect, clinical relevance, safety profile, uncertainty, and feasibility of additional trials. Benefit risk assessment must be contextual, especially for serious or life-threatening diseases. Common pitfalls include post-hoc analysis, weak external controls, poorly defined endpoints, inadequate blinding, missing pre-specified rules, and underpowered studies. To wrap up, FDA's draft guidance reinforces a flexible, science-driven approach to demonstrating effectiveness. One pivotal trial plus strong confirmatory evidence is a viable pathway. FDA is increasingly supportive of innovative trial designs and real-world evidence as long as they are rigorously executed. And for rare diseases, FDA offers methodological flexibility, but the evidentiary standard remains unchanged. The most important takeaway: engage FDA early and often, especially through Type B and Type C meetings. The bottom line: FDA's framework is flexible, science-based, and designed to support modern development, but rigor still matters. If you found this helpful, subscribe for more regulatory breakdowns. Thanks for watching. Have a wonderful day.